All right, so not all lipids are composed of glycerol and fatty acids. Okay, some of them are a little different. So we know triglycerides are composed of glycerol and 3 fatty acids. All right, but there are others that are composed of other things. Okay, if we use the basic structure of a triglyceride, glycerol, 3 carbon molecule, and 3 fatty acid side chains. And we replace one of those fatty acids, take it off, and we put in place of it a phosphate group. Okay? That is essentially, a phospholipid. Okay? So what it looks like is, here's your carbon, 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 glycerol molecule, right? And fatty acid side chain. I'm not going to draw all the carbons out. Fatty acid side chain. Okay? And instead of having a 3rd fatty acid side chain right here, we're going to put in a phosphate with oxygens around it. Okay? The unique thing about this now is that what do we know about these fatty acid side chains? What do you know about this? What type of bonds are between these carbons? Covalent, but they are non-polar. Okay, that is why triglycerides and phospholipids have this, makes up their hydrophobic nature of this lipid. Okay? Before Max throws his water bottle at people.
So excited.
Okay? So this side of this phospholipid, this side is hydrophobic. And the reason why it's hydrophobic is because of the non-polar covalent bonds between the carbons. Okay? Well, what about the other side? What about over here? Why is it hydrophilic? Wait, stop and think. If I'm telling you that the phosphate is hydrophilic, meaning it likes water, give me a reason why it likes water. It is polar. So what does it mean when it's polar? It has to be unequal sharing, charged. Okay, there's a charge to it. Okay, and so there's a charge molecule. So you'll see we kind of draw a phospholipid with a phosphate, a polar phosphate head, and non-polar fatty acid tail. So it kind of looks like, I'll draw it like this usually. Okay? So the good thing about this is that your cells are made up of mostly what? Water. So because the phospholipids have one area, the phosphate head, that likes water, and the non-polar region over here that doesn't like water, what does it end up doing? It ends up orientating itself towards, in this fashion. Okay, forming a bilayer. Anyways, so you have this bilayer that's present, where you arrange the polar heads towards the water, and the non-polar tails are towards each other. This is called a phospholipid bilayer, right? And you have a hydrophilic region, which is out here and in here, where the polar heads are, and a what region that's in here? Hydrophobic. Okay? Very good. This is the basis for cellular membranes. Okay? And then from there, we're going to add in proteins in order to move things in and out. and we're going to add in some carbohydrates and some other lipids so that we get a fully functioning cell membrane. But this is the backbone of it, okay? Now, what is a function of a cellular membrane? Transport, what else? A barrier, move things in and out of the cell. So transport involved, right? What types of things, just in this phospholipid bilayer, what types of things can pass through? Or what types of things cannot pass through? There we go. Water, okay? And water is a, what type of molecule? Polar. Well, give me another example of a polar molecule. Someone said something back here. I heard something. Give me another example of another polar molecule. Or maybe not polar, but something else that won't go through a phospholipid bilayer. Okay, proteins because they're probably too big. Okay, what else? Without specialized proteins inside the membrane, okay, things like sodium ions, chlorine ions. Why won't those things go through? Because they are charged, they're polar. Okay? So anything that's charged or polar won't go through because of the hydrophobic nature of the phospholipid bilayer. Okay? So you have phospholipids, you have your two fatty acids and a phosphate group that are attached to glycerol. The two fatty acids are your hydrophobic regions. Your phosphate group is your hydrophilic region because it is polar. All right? And this is your major constituent or major thing that makes up cell membranes. Okay, so this is what it looks like. This is a picture from your book. Here's a space-filling model. Tell me something about this right here. Tell me something about this fatty acid right there. It's unsaturated. Why is it unsaturated? What about that picture tells me it's unsaturated? There's less hydrogen than there could be. Okay. What else?
There's a double bond for the little chain part.
Right? So there's a double bond right there. Okay? Now, what about this picture? So if I didn't give you, let's say I didn't give you this, so you couldn't tell. I just gave you the picture. What if I just give you the picture? Can you tell from there?
Is it because it's bent?
It's because it's bent. Because it's bent. The tail is bent. That's where the double bond is. There's not a saturation of hydrogens. Okay? And because it's bent. So you could tell just from the picture that that is a unsaturated fat. And what type of lipid is this? It's a phospholipid. Okay? The unsaturated fatty acids that are attached to phospholipids prevent the phospholipids from becoming too close together and becoming too dense in one area, becoming too packed together. I'll draw it out like this. So if I have phospholipids with straight tails, okay, I can pack them in pretty tight, right? But if I have phospholipids with bent tails, I cannot pack them in as tightly. Okay? This is important because it allows for membranes to remain fluid. Cell membranes need to move. Do you agree with that statement? Okay? Cells need to move. So cell membranes need to be adjustable. They need to move with the characteristics of the cell itself. So membranes are fluid. Okay? So they will move things around accordingly. So when phospholipids are added to water, they do something what's called self-assemble into a bilayer. Why? Hydrophobic tails, hydrophilic heads. All right?
So the ones with thin tails are unsaturated.
Yes. Okay? This feature results in your bilayer arrangement found in your cell membranes that looks like I've been drawing. Right? That's your phospholipid bilayer. Questions on phospholipid bilayers and the structure of phospholipids in general. Okay? We're going to come back to that when we talk about plasma membrane next week. Grace.
So it has one saturated and one saturated, unsaturated section?
Yes. There is a whole, we're generalizing a phospholipid.
Okay.
Okay? So I give you a generalized structure. There are tons of different phospholipids. Some that have two saturated fatty acids, some that have two unsaturated fatty acids, and some that are mixed between the two. So there's a whole host of them that make up the cell membrane. We're just giving you a generalized one. And for this case, when you see the bent, it is one saturated and one unsaturated. Okay? The other type of lipids that are important are steroids. Okay? The reason why steroids are characterized as or categorized as lipids because their skeleton is carbon-based, all right? And they are hydrophobic. Steroids do not or are not composed of glycerol and fatty acids, okay? Like phospholipids and triglycerides are. Instead, steroids are built upon the cholesterol backbone. This is characterized, or the structure of this is what we call 3 rooms and a bath. Okay, that's what I learned when I was sitting in your seats. Okay, basically you have 3 6-member rings and a 5-member ring. That makes up the structure, the backbone of cholesterol. So they're fused rings as well, meaning they're attached to each other. Okay? And it looks something like that. That's the back room. Your 3 rooms, and then your 4th is your bathroom. It's a 5-member ring. Okay? That's the carbon backbone. Okay, so when we're going to talk about cholesterol, it's going to have this structure and then some different side groups off the end. Okay? And then go from cholesterol to testosterone is going to change some bonds inside here and the side groups. And then going from testosterone to estrogen, the side groups change again. And if we want to look at progesterone, the side groups change again. Okay? But they all have this structure. The carbon skeleton stays the same. The bonds between them may change, and the side groups may change. But that structure stays the same. Okay? And that's cholesterol. Okay? Cholesterol is also important in membranes because it wedges in between that hydrophobic layer of the fatty acid tails and prevents those phospholipids from becoming too closely packed to one another. Okay? So that helps maintain fluidity and strength of membranes. All right? Why would cholesterol be found in the hydrophobic region and not the hydrophilic region of the cell?
Because it's not polar.
Because it's not polar. Because it's a lipid. It's hydrophobic. That's part of the nature of it. Very good. Okay? So, in the lab, I want to look at something. I want to look at the effect of testosterone treatment on cells growing in a dish, right, that are going to become muscle. So there's special cells in your muscle that will help grow and produce more muscle. I want to look at how testosterone affects that because we know that that happens, right? Okay, so I want to look at it in the lab, but the way I grow cells in a lab is in an aqueous solution of glucose and everything else, right? And the cells are basically an aqueous solution, correct? All right, so now I order testosterone and it comes in a powder. Is it going to dissolve in that aqueous solution? Someone's shaking their head no. Why? Because it's a lipid. It's hydrophobic. So what do I got to do? It's a powder. Oh, crap, I'm screwed. My thesis isn't going to work. My application for tenure is going to get denied. I'm going to have to find a new job. What do I need to do? Not everything is going to be soluble in water. In this case, I have to find another solvent. This is a hydrophobic compound, okay? So it's soluble in ethanol. So I would have to put it in ethanol, solubilize it, get the concentration, and then I could put it on cells, and it would get into the cells from there, okay? But once it's in ethanol, how do I know it gets into the cells? Does it get through? Does it get through on its own? Why would this cholesterol-based steroid, such as testosterone or estrogen, how do I know it gets through? Does it get through the cell membrane? How many of you say yes? Okay, 5 or 6 of you. Gina says yes. Why? Because it's nonpolar. Okay? So the nonpolar nature of this allows it to slide past the cell membrane. Okay? This is hydrophobic too, and it can go through. Right? A lot of nonpolar molecules, they go through, they can diffuse, and mean they passively cross the cell membrane easily based upon their structure and their non-charged nature. Okay, so here's the backbone of cholesterol. There's your three rings, your 3 rooms in a bath. Okay? Now, like I said, we're going to change all those subgroups, all these subgroups here. These are going to get changed. Look how complicated that one is. Those are going to get changed. We're going to go from cholesterol to other things. Okay? When we go from testosterone, it's a different group. We go to estrogen. There's some changes there, and it does something else. Okay? Obviously, estrogen does something very different than testosterone. Correct? Yes. And estrogen and testosterone do something very different than cholesterol. Yes? Structure and function. Okay? The side groups change, the structure changes, the function changes. Okay? Questions on lipids. So 3 types of lipids. What's the first one? Triglycerides. Okay, and they can be saturated or unsaturated, the fats, glycerol, and fatty acids. Then we have the second type are phospholipids, and the third type, steroids. Okay? Questions on lipids. So we have 2 more types of macromolecules to cover, and that includes proteins and nucleic acids. All right? Proteins are the most diverse in terms of function and structure of the macromolecules. All right, we talked about how these macromolecules are built. How are they built? How do we combine monomers or subunits? Through what? Dehydration synthesis. Okay? And that is Also true here. All right, so proteins account for more than about 50% of your dry mass of the cell. They're found in ribosomes, they're found in the nucleus, they're found throughout the cytoskeleton, parts of the cell that we'll talk about next week. Okay? Their functions include defense, antibodies, include storage, okay? They include transport, movement of things in and out of the plasma membrane and through the cell. Cellular communication, such as hormones, okay? Movement, the cytoskeleton, and structural support, the cytoskeleton, okay? They are a very diverse type of group, okay? Here's some examples. This is in your book. You have enzymatic proteins, enzymes. We'll talk about them in detail. You have defense proteins, such as antibodies, storage proteins, right? Storage of amino acids, ovalbumin, which you've used in lab, right? That's a protein for storage. Hormonal proteins, such as insulin, is a hormonal protein. Contractile motor proteins, actin and myosin, for muscle contraction. Structural proteins, collagen is the main one. Collagen is the most abundant protein in your body. Okay? Receptor proteins, acetylcholine receptor, signaling molecules, so you can relay messages, transport things in and out. Things are either charged, too big, too complex to get through the membrane on their own, go through a protein that's embedded inside the membrane, and so on. All right? Function, very different than what we've talked about in terms of carbohydrates and lipids, right? Carbohydrates and lipids are basically, carbohydrates are for cellular energy, lipids are for membranes and the such. Okay, also used for energy storage. Is that there again? We can skip those. All right, so one group of proteins is enzymes. All right, enzymes work as catalysts, and you'll be talking about this in chemistry, all right, about rate of reactions and how fast they go, whether or not they go faster in the presence of an enzyme or not. But what this does, the enzymes in your body allow reactions that could occur without the enzyme, but allow them to go faster. Okay, by lowering activation energy required of that chemical reaction. Okay? Now the good part about this is that they act to speed up this reaction, however, they're not consumed by the reaction. They are not, they are reusable. Okay? This slide is meant to tell you a difference in vocabulary. When we talk about, I will use these sort of interchangeably, okay? But there is an intuitive difference between a polypeptide and a protein, okay? Proteins are composed of what? Amino acids, okay, that are linked together. So the macromolecule protein is a collection of, or a string of amino acids that are amino acid subunits that are linked together through dehydration synthesis. Okay? And as you add amino acids, you produce peptide bonds between amino acids, and now you have a string of amino acids, which they call a polypeptide. Okay? That polypeptide may or may not be functional in that structure. Okay? A protein is different from a polypeptide. Okay? All proteins are polypeptides, but not all polypeptides are proteins. Okay? The difference is this word right here. Is right here. Biologically functional molecule. Okay? A protein denotes functionality. It can do the job it is intended to do. A polypeptide may not be functional, may not be in the right structure for it to be functional. Okay? It might not have the right interactions for it to be functional. Okay? Example. Insulin, what we just talked about, is a functional protein. Okay? In the cell, inside the cell, beta cells of the pancreas, it is produced in what we call a pro form. Proinsulin. It is not active inside the beta cells. But as it's released from the beta cells into the bloodstream, a part of the protein is cut off, and now it becomes a functional protein. So proinsulin is a polypeptide. Insulin is a protein. Okay? Do you understand the difference? The main difference is right here, the functionality. Okay? If it's not functional, it is not considered a protein. Okay? A lot of times you will use them interchangeably. When I say protein, assume a biologically functional molecule. Okay? How do we get to that point? How do we go from a chain of amino acids, or amino acid 1 floating out here, amino acid 2 floating out there, amino acid 3 just kind of random? We have to link them together, right? And then from there, they have to be linked in a right sequence. Then they have to assume their correct structure. Because structure is directly related to what? Function. Okay. So, amino acids. They are organic molecules with a distinct structure. Okay? They all have what we call a central carbon. So here's a carbon. How many bonds can carbon form? 4. So 1, 2, 3, 4. Okay? This is the structure of amino acid. Every amino acid has one end, which is an amine group, and the other end, which is a carboxy group. Okay? So an amine group means what? You remember? NH2. So NH2. Carboxy group means what? COOH. Right? So that's also C double bond O, OH. Same thing. Okay? So this end is the amino terminus, and this end is the carboxy terminus. Okay? Right here's your central carbon, and there's another hydrogen right there. Okay? So all amino acids, all 20 amino acids, share this structure. So why do we have 20 different ones? The difference is right here in this R group. Okay? That R group is the variable. It could be as simple as a hydrogen, as complex as a ring structure. Okay? Long chains, whatever it might be. But that R group determines whether it's glycine, one amino acid, versus isoleucine, which is another amino acid, or tryptophan, or tyrosine, whatever it might be. Okay? The difference is that R group. So here it is, central carbon, carboxy or carboxy terminus, and your amino group on the other side. Your side chain, the R group, is the difference between amino acids. Here are your 20 amino acids. No, you don't have to know them. No, you don't have to know the structures. Okay? This is meant to show you what they look like. They could be pretty complex, such as tryptophan with double ring structure. Here's glycine, which is your hydrogen. Right? Then we go to a methyl group, a 3-methyl, a valine, right? And most of those these are all non-polar side chains. Why is it important that we distinguish between non-polar and polar and acidic or basic side chains? What's that?
See if you get out of the cell.
Well, it's to see if it can get in and out of the cell, yes, but what are those properties going to do? Affect the function. Because ideally, we're gonna produce a protein that's gonna have a long string of amino acids that each one of them is gonna be one of these 20, right? And when we talk about the overall structure of the biologically functional protein, these side chains are going to determine that structure, which will then determine the function of the protein. Okay? Like I said, you don't need to know them, but you have to understand that the overall structure of the protein is a result of these R groups. Okay? So, polypeptides. Your amino acids are linked together by a special bond called a peptide bond. That's why we call a string of amino acids, a polymer of amino acids, a polypeptide, because there is multiple peptide bonds. Now, we can distinguish between what polypeptide is. It can range in length from a few to more than 1,000. Okay? So, it could be a long chain of amino acids. Is that a functional protein yet? No, right? If it was a functional protein, I'd call it that. Polypeptide is just a string of amino acids. All right? Each polypeptide, each protein that you have has a distinct linear structure. All right? A distinct sequence of amino acids that are put in the right spot. Okay? With a carboxy terminus and an amino terminus. And that's because each amino acid has a carboxy terminus and an amino terminus, right? So here, if I'm going to join amino acids together, here's amino acid 1, 2, 3, 4, 5. This amino acid has a NH2, well, NH2. And this amino acid has a C double bond O OH. So you have your amino terminus and a carboxy terminus. Okay? Because we're just going to link these amino acids, 1, 2, 3, n number of amino acids in succession. We're going to produce those peptide bonds between them, right, in order. So, and this is done through dehydration synthesis. So here is 2 amino acids, okay, right, side chain, side chain. Here's the carboxy terminus of the end amino acid, and the amino terminus of the next incoming amino acid. Water comes out, peptide bond is formed, and you end up with a polypeptide that has an amino terminus and a carboxy terminus. Where's the next amino acid going to go? Right here, right? So you're going to add amino acids at the carboxyl end, at the carboxy terminus. Okay? The sequence of amino acids and how they are arranged, amino acid 1, 2, 3 for whatever protein, is important. That is your primary structure of the overall protein. Okay? So we're going to talk about levels of structure over the next 2 days. Okay, today and on Monday. And there are 4 levels of protein structure. Okay? The primary level is the number and sequence of amino acids. Okay? So protein A has 30 amino acids that go 1, 2, 3, 4, 5, 6, 7 to 30 in that range. Protein B also has 30 amino acids, but instead of 1 through 30, right, it has it's amino acid 1, 6, 5, 4, 2, 10, 11, 7, different order. Okay? It's in a different order. So instead of going glycine, alanine, proline in a protein B, it goes proline, valine, tryptophan in protein A. All right? So it's different which amino acids go in that order is important. Okay? I have not stressed this enough. A functional protein consists of one or more polypeptides, precisely twisted, folded, and coiled into a unique shape. And that shape determines what? Function. Okay? The shape of an enzyme is directly related so that it can recognize the substrate. Give me an example of that. What did we talk about on Wednesday? That is an example of an enzyme that recognizes a certain substrate. Not estrogen. What? Glycogen and cellulose. The fact that we as humans cannot recognize and break down cellulose, we don't have an enzyme that recognizes that substrate. Okay? We don't have an enzyme that's folded in the right way to recognize cellulose as a substrate, so it gets passed through the body as undigested material in fiber. Okay? So here's what you see. You may see ribbon molecules, right? A structure or a space-filling molecule of a overall protein. What you see here on the ribbon structure, you see a couple coils, right? Those are alpha helices. That is secondary structure. Okay? There's up here, and kind of goes back and forth like that. That's a beta sheet. That is secondary structure. So parts of the protein will fold in a certain way, and then it will fold on top of itself again, making a globular protein. All those folds are secondary and tertiary structure. And then beyond that, we could have multiple subunits of protein, of peptides come together to form a functional protein that is quaternary or fourth degree structure. Okay? So your sequence is determined genetically, and it will eventually lead to the protein's three-dimensional structure. Okay? I made a point of emphasis about the R groups because the sequence of amino acids is primary, but the sequence also determines where those R groups are going to be. Okay? So that we have a tryptophan which has an elaborate ring structure. R group is not maybe next to a glycine as opposed to next to another amino acid that has another ring structure. Okay? Because of how they interact in space. Okay? Structure and function related. All right? So here's an antibody protein. Look at that structure. And a protein from a flu virus. Look at that structure. Why is that important? What's that going to do for you over here? How many of you have gotten a flu vaccine this year? Why did you get a flu vaccine? Because my mom maybe. No. Right? Why is it important to get a flu vaccine? What does a flu vaccine do for you? If you build antibodies up against the flu virus. This is a flu virus. Here's an antibody. This is what your body's producing. It's going to react to this flu virus so you don't get sick or you don't get as sick as you potentially could. Okay? Structure and function. Structure here. You're producing this structure to recognize this structure, and it will bind to this functionality. So now this doesn't infect your cells. Okay? Oh, no, I didn't want to skip that one. So we have 4 levels of protein structure. Okay? Proteins are diverse in terms of functionality and structure, but they do share 3 different levels of structure called primary, secondary, and tertiary structure. The 4th level, quaternary structure, is not present in all proteins. Okay? It consists where you have different parts, multiple separate polypeptide units that have their own shape, but they come together to form a functional protein. Okay? So actually more than one polypeptide chain. Okay? Questions on that? We'll stop there. We will pick up with this on Monday. We'll finish proteins and nucleic acids on Monday. Okay? for your studying for your exam, I will post a study guide this afternoon. The study guide is just a list of topics which will be covered, which we have covered, and which will be on your exam. Understood? Have a great week.